Hello, students. Do you hear me? Hello, students. Give me a sign. Are you there? Okay, students, are you there? Somebody answer me. Are we going to have a lecture?
Hello,
Hello,
doctor.
doctor. Hello, are we going to have a lecture or not? Nobody is answering me.
I think yes.
Say, please tell it again, I didn't hear you.
We cannot
We
hear
cannot
you
hear
very well.
you. Don't you hear me?
Not very well.
Oh, so what is the reason? I don't know. Here with me, I have the microphone and everything.
I don't know. It is not very clear, the sound.
So are we going to start or waiting for more? Let's have several people appear again. Please tell me, are we going to start? Do you see at least
Yes, we can see.
the presentation?
Yes, yes.
Do you think we should start already?
Yes, we can start.
Okay. So if you don't hear me or something happened, please call me again. Okay?
Okay, yes.
Tell me so to see what happens with the slides here. Oh, don't tell me. Where are these slides? Okay, the topic of today's lecture is the relationship between the malignant cells or cancers and the immune system. This is a very complex and difficult relationships because but it's all processes of the immune system, such as antigen presentation and recognition of tumor antigens, activation of antigen presenting cells, which might present these tumor antigens, T cells mobilization. Also, T-cell interactions with the tumor cells are quite specific here, and we expect this immune system to disrupt the tumor cells, but it's not always happen. Let me find it here. Okay. So the development of the cancer is a process which is connected with the multiple genetic and epigenetic phenomenon. Several clues of the achievement of the cancer immunology. Now it's known that the human tumors appears very heterogeneous, different cells with different specificities in the same tumor, which makes the relationship with the immunology more complex than it was expected. Complex relationship between the tumor or cancer and the microenvironment and the milieu around the tumor cells, which is very important because it gives or deliver oxygen and food substances for the tumor cells. And yet the interesting thing in the last several years is that the hypothesis for the stem cells is concerned also with the cancer stem cells, which are the original for development of different malignant tumors. So, which make a different paradigm to change the therapeutic paradigms, which means that um, radical eradication or radical treatment of the specific tumors is means to eliminate the tumor stem cells, which is very a different side to the therapy. So several predisposition factors are connected with the malignant development, and they are connected with the factors which are inherited immune deficiency or acquired immune deficiency during the life, and this is connected with defects in the immune surveillance, leading to long-lasting cellular proliferation of tumor cells. And this is confirmed such situations which are inherited immune defects, inherited other genetic chromosomal defects, and also acquired such one. One class known is that the environmental factors such as viruses, chemicals or physical factors are important to, and they produce mutation effects which are in the origin of tumor cells. We'll see more after that. And as I already said, inherited or acquired chromosomal defects are connected with the increased cellular vulnerability, which means that the cells are easy to destruct. It, it's the relationship between the chronic inflammation and the carcinoma is well established, and one of the ways this is happening with the intercellular second messenger pathways, which there is the same mechanism such as STAT3 uh, second messenger, which is situated in the tumor cells and also in the immune cells. An interchange between these cells might appear because when STAT3 is activation of the tumor cells, might promote cytokines and induce tumor growth, which means it's connected with, because these STAT3 molecules are connected with the cytokine production, this might produce pro-cancerogenic uh, cytokines. And on the other hand, the STAT3 activation of the immune cells might depress the tumor specific immune responses and inflammation, as well as in inflammation. And there's a pro-inflammatory cytokines such as interleukin 6 or 11, which might induce uh, inflammation, but they might be interchange dysregulation, and they might become from inflammatory toward cancerogenic. So production of cytokines uncontrolled or dysregulated might induce tumor growth. And this is very important because this is one of the mechanisms by which the inflammation, chronic inflammation, and infections might induce or take part in the creation of the carcinoma cells. Nowadays, concepts for carcinogenesis from immunological point of view is so-called concepts of 3E, which are processes connected with elimination, equilibrium, or escape. These three E's are in the basis of this relation with tumor called cancer cells and malignant cells and immune system, which means which leads to immune editing. This is a process which is connected with loss of the sensitivity toward from immune system toward tumor cells. So let's have a little bit more proper look about this. Normal tissue, there is no tumor cells, And there are processes which are connected pro-cancerogenic, which is chronic inflammation, as I already mentioned, inherited genetic mutations, and other viruses, often viral infections, which might induce cancerogenesis to make normal cells toward tumor cells. But in organisms, there are mechanisms which are connected with the fighting this pro-cancerogenic phenomena which are all the time in the cell. And this is repairing, DNA repairing, senescence, or apoptosis inhibition. These processes are connected as a counterpoint against this tumor creation uh, phenomenon. So, but when cancerogenesis goes, it, as you see, all the time it is possible to some part of the cells or some protein mutate and make these cells as a cancer one. And these cancer cells are all the time under the surveillance of the immunity, different phenomena of immunity. And down there you can see how many immune cells are taking part in this immune surveillance, which is macrophages several times as an antigen presenting cells, gamma delta T cells, NK cells, NKT cells, CD8, CD4, and so on cells. These uh, cells, immune cells, acting in concert as a health phenomenon is to eliminate or destroy these tumor cells, so to bring back the tissue in a healthy state. But in some cases, and this uh, um, phase is called elimination, effective elimination of the tumor cells from the immune responses. But sometimes there is not so effective immune cells, and some tumor cells, as I told you, there is a high rate of heterogeneity or appearing of different tumor-specific cells in the tumor mass. And if this process appears, not all tumor cells are recognized and effectively fight by the immune cells, and but still the immune response is effective, and not very much cells are appearing. The tumor is staying in the phase of tumor cells, and these processes are vice versa, and this um, phase is called equilibrium between the tumor growth and immune system and the phenomenon of immune system. But sometimes the tumor cells became very big, and this phase escaped from immune control. So we go in so-called escape of tumor from the immune surveillance, which these immune responses are surveillance. We'll see more. A lot when these tumor cells or growth became a bigger one, or different new types of heterogeneity of tumor cells appear, a lot of mechanisms are switched on, which lead to suppression of the immune system. As you see, the suppression of effective presentation, the suppression of unusual cytokine production, and other mechanisms are switched on, which leads to escape of the tumor growth from the immune surveillance. So let's see a little bit more. Which are the mechanisms, as we talked, just to remind, which take part in this immune surveillance or the effective elimination phase? These are mechanisms of innate immunity and also acquired immunity. Innate immunity is the face the first cells which face the first appearance of the tumor cells. These are NK cells and macrophages, which, when they are activated, NK cells lyse the tumor cells, and macrophages produce different factors or biological active radicals, oxygen radicals or TNF, which also intend to lyse the tumor cells. But when it is not enough, the other mechanisms of acquired immunity are introduced, more antibody-mediated uh, cytotoxicity from the macrophages or NK cells. There are also very active processes. Immune system, when recognize the tumor antigens, produce also antibodies, which in some cases might be effective, but this is not a very effective mechanism. Humoral mechanism is not effective to lyse or to kill the tumor cells. And the strongest mechanism is so-called T cell, CD8 positive cell cytotoxicity, which make the tumor cells rise. But they include the fight. Went in the fighting quite late. There is time needed to these cells to be activated. In the same time, tumor, as I told you, tumor cells, when they are more bigger, they produce the effective suppression, which suppress all or some of this mechanism, and and this is the moment when the tumor cells escape from the immune surveillance. We'll see more about this. The most effective mechanism is rising tumor cells from NK cells or by CD8 positive T lymphocytes, and this is the way the elimination of tumor cells is induced by the immune system, the most pronounced way. Different mechanisms of this cytolysis of tumor cells from T lymphocytes are known nowadays, and one of them is extra cytosolic release of granzyme and perforins. These are proteins which directly lyse tumor cells. Another mechanism is by expression of the ligands or receptor in the T cells or tumor cells, and one such pair is TNF receptor, which are recognized by TNF secreted cytokines secreted by T cytotoxic cells. And another pair is expression, as I told you, of different mechanisms. So-called fast ligand is expressed on T lymphocytes, and fast ligand, receptor is on T lymphocyte on a tumor cells, express fast ligand, and the result is apoptosis of these cells. So different mechanisms in different situations are released by these cytotoxic NK cells and CD8 positive cells. I told you in certain in a moment how to say in a certain moment of the process of tumorigenesis, the equilibrium is established, and this is when the tumor cells, the tumor itself is not growing very fast. It keeps a small micro volume. It's a small volume, and this is when the immune systems, as you see here, CD8, CD4, NK cells, gamma delta cells, and so on, are effective, and they are able to keep single or single tumor cells, but in some moment this heterogeneity of the tumor increased, and they escape until last they escaped from the possibility or ability of immune cells to lyse them. And this is the moment I told you when the escape, the third E, is the escape of tumor cells from the immune surveillance, and this is because of reduced immunogenicity of some tumor antigens. This reduced and this which this make the tumor cells and tumor antigens unrecognizable, or the reaction they induce is not effective to lyse the cells. And there are several mechanisms here are notice there. What is there might be cytokines, there might be activated around presentation from the antigen presentation cells, dendritic cells, immature dendritic cells induce tolerance but not activation, and a lot of mechanisms, cytokines also, which suppress suppress the immunity, and it is not effective, this recognition and equilibrium. So then in this moment the escape of the tumor and uncontrolled growth of these tumor cells is the result of this escape of immune regulation. And this process, as I told you, not only from the wrong recognition we will see, but it's very important, these suppressor processes and induced suppressor cells, which are induced by the tumor itself. Which what that means? There are a lot of identified suppressor cells, and the first one, as you see, we already talk about this. These are so-called CD4 positive, CD25, and FOXP3 regulatory T cells, Treg cells. We spoke about them in autoimmunity, where these cells are reduced. But on the other side, in tumors or in cancerogenesis, these cells are increased as a number, as an activation, and they suppress nearly all other immune processes, which we'll see a little bit later. Another big group which was of suppressor cells is so-called myeloid-derived suppressor cells. These cells were identified the last 10 years, and it appears that they are very effective in suppressing the immune system. They are also induced by the tumor cells, and they are specific for these tumors. Interleukin 2 producing immune cells, these are usually B lymphocytes, but also T lymphocytes, which express suppressive action onto the immune system. And another NKT suppressor cells, which are very, very important, and they're effective in suppressing the immune response. These slides I already showed you. Just to remind, these are T regulatory cells. They are very central cells in regulating the immune response. For tumor cell in cancerogenesis, these cells are increased as a number, on the contrary of autoimmune situation where these cells are reduced. And a lot of molecules which are important and well known, proven to induce suppression on the tissue, on the immune system, also another immune cells, secretion of cytokines, expression of other HLA, VR molecules. They are antigen specific. They recognize tumor antigens through the TCR receptor, that T cell receptor, alpha beta, but also gamma delta receptor, and so on. Also, these cells are able to interact with so-called toll-like receptors, which are very important in the innate immune phase of the response. And these Treg cells express their action onto all processes of the immunity. But not only cells has this suppressive effect. A lot of cytokines which are produced by this T regulatory or induced by tumor cells itself produce cytokines such as tumor growth factor beta, interleukin 10, and growth factor also another. And all these two cytokines express very strong suppressive effects on the nearly all mechanisms of the immunity, which means T cell recognition, T cell growth, differentiation, B cell activation, cytotoxic potential of CD8 positive cells, and suppressive effect, and so on. So these as suppressive cells are important, and they call it T regulatory cells, but also cytokine or humoral factor are very important in suppressing this immune phenomenon. And why it is important? Because this is the reason that the tumor growth escaped from the immune surveillance. And this escape of the immune surveillance might due to defective recognition of the antigen, which is connected with, you see, expression of the antigen on the tumor cell surface must be done in HLA class 1 or molecules. But tumor cells is very clever, and it's thought not to express HLA class molecules 1 and 2 also, and we'll see why. And this antigen, nevertheless, they are on the surface of the tumor cells. They cannot be recognized by T cells, and there is no immune reaction. So we call this defective antigen presentation and recognition. As I already told you, expression of ligands and receptors are the reason when the fast ligands are interchange, they kill T cells, not tumor cells, but T cells usually. And as I told you, the different other molecules are mask the tumor antigen. Why it's so important these tumor antigens to be recognized by the immune system? Because of these reactions depends how this surveillance of the immunity over the T cells should be explained. But as I told you, very much very important is this immune suppression, which is induced from the tumor cells, and it might be T cell induced, T regulatory cells, and cytokine. The important roles of the for the escape of the immune response are genetic instability of the tumor antigens. And there are two phenomena called shedding of the tumor antigens and antigen modulation, which are very characteristic for the tumor cells. Normal cells has a lot of proteins which are expressed on the surface or inside in the function, and they're stable and they're not changing, but not in the tumor cells. In tumor cells, these tumor antigens, might be produced, but when they make this process shedding from the surface, and when these shed antigens are in the blood or in the lymph, they became humoral, but they induce tolerogenic effect toward these tumor cells. So this is mechanisms of the tumor cells shedding its antigen to induce suppression over the immune system. And antigen modulation is the other very characteristic process, which means that tumor antigens might very often might disappear from the surface of tumor cells, and this way they are not visible for the immune system, or they disappeared as an antigen, but also disappeared HLA class 1, which is prone to present these tumor antigens on the surface. So this is called antigen modulation. This decreased expression of HLA class 1, as I told you, is a process which was discovered the last 10 years maybe, or less. And the reason for this, and as I told you, it's very important that then it makes the immune response not effective because the antigen is not recognized by the immune system. And the reason for this is secretion of the cytokine TGF-beta, which is the most strong suppressive cytokine on the whole immune phenomenon, as I showed you in the table before. So this secretion induced not effective. It affects one of the chains in the so-called CAPS, which takes the HLA class molecule and express it on the surface of the cell in the connection, in the context with the antigen. So this TGF block expression of this CAP chain, and the whole process is not effective. And this is a widespread phenomenon. It was discovered for carcinoma, different carcinomas, lung, colon, breast carcinoma, carcinoma of the tongue, cervix, and so on. So this is a valid process for most, maybe all tumor localizations or types of carcinomas. So the uh, development of so-called tumor immunology is connected very strongly with the identification of the antigens. Tumor antigens might be specific for the tumor, might be connected with the other factors, and there is a presentation that they might be viral antigens which infected and induce tumor cell transformation, and the viral antigens might be expressed on the surface. But for humans, I will tell you, very few antigens are expressed on the surface. Other antigens might be expressed from viral antigens, which are so-called oncofetal. The most often oncofetal antigens, which are expressed on the surface, and we'll see more about so-called tumor markers on the cells. There is a great family, a big family of antigens which are identified in tumor cells in skin carcinomas or lung carcinomas, and these are mutants specific for the tumor's antigens. So well known here to uh, antigens P53 connected with the carcinoma, and loss of organ specificity, as I told you, NHC class, and so on. So different antigens must be or shall be expressed on the surface. Different the detection of tumor antigen and recognition is important because if the tumor antigen is presented, or if this is a tumor cells and it's presented on HLA and in the hood and recognized by T cells, then T cells might be activated. But it's absolutely necessary to have this second signal, which is important to uh, transduce the signal to the nucleus of T cell to make an activation. But tumor cells is clever, and they do not express some of these additional adherence antigens. So the final results of this antigen recognition might be normal, but the result is tolerance or energy, not activation nor active immune responses. The expression of T inhibitory molecules and cytokines also induced, you see, there's molecules, all immune responses are uh, not only cellular recognition, but also secretion of different cytokines, which might be activating or tolerating or suppressing the immune response. So the results, which some of it, if say this is interleukin 10, if it is secreted, it suppresses quite strongly the immune responses. And another situation when the immune cells do not produce this HLA expression, and this tumor antigen might be also not expressed on the surface. Tumor antigens might be classified as a specific one, tumor-specific antigens. They are characterizing certain tumors. They are more characterized in so-called experimental tumors because you know what you're looking for there. In viral-induced tumors also might be identified such antigen and chemically induced tumors. But these situations are characteristic for the experimental tumors, mostly in animal laboratory animals. In human, it's very difficult to identify such an antigen, characteristic for viral antigen, or for very few which are identified as a specific for certain for melanoma. Most studies are done on melanoma too. And there is another group of so-called tumor-associated antigens, which are expressed in normal cells and in a higher level in tumor cells also. So these are the group of so-called tumor markers. I think you met these uh, definitions in some clinical and internal medicine. That's why, and we will have a little more work about this. Usually, most often, these tumor markers, which appear in normal and in tumor cells, are originating for so-called oncofetal antigens, and they are best characterized. I'm not going to go very deeply in this, but we will see several groups of antigens here. If we connect the normal gene production, so it means that these are market tumor antigens which are results of functioning or over functioning of normal genes in the cells. And these are case when the tyrosinase is expressed on the surface of tumor cells. And this is a well-defined tumor antigen, specific tumor antigen in melanoma cases. Here there is the big group of oncofetal antigens, and these the most known are alpha fetoprotein or carcinoembryonic antigens. They are originating from the liver cells or from fetal other epithelial cells, and they are usually the most used factors, two of them, of course, in as a tumor markers. And the other type of normal antigens are when mostly in hematological malignancy, where differentiation antigen persists longer time and in periphery in this lymphosis or lymphoproliferative diseases, where the cells are carrying some of this early in development antigen, differentiation antigen. And here the group is mutation antigens, which are abnormal mutation protein, which are might due to chromosomal translocation or dot mutation, and here these are these viral antigens on the surface. These groups are known for what is important here, that these different antigens are usually nowadays used as a target for so-called target immunotherapy in cancer. We'll have a little bit more. So these are in human, the viral antigens which are identified up to now. You see, these are not very much identification. Epstein-Barr viruses is characteristic for Burkitt lymphoma. It's considered a nasopharyngeal carcinoma. It's considered as a inducer of this uh, carcinomas, and that it is proven in humans, it's expected in humans, but proven in different models. So these are one of the antigens, viral antigens, which might be identified. Human T lymphocytes virus attacks T cells, and it's characteristic for T cell induced. It's considered possible to induce T cell leukemia. Human papillomavirus viruses is popular for expectation to induce carcinoma of the cervix, from cervix, and it affects infect skin or mucous cells, and this is the way it take part in carcinogenesis. Human hepatitis B virus and hepatitis C virus, especially for hepatitis C virus, it is considered to take part in pathogenesis of primary liver carcinoma. Also, there is a very strong suspicion that human herpes, some of the herpes virus might be affecting or infecting T cells, and the reason for not identified carcinoma up to now. So, as usual, HIV virus is considered for Kaposi sarcoma. The thing is that you see not very much identified in human possibility to induce uh, increased immune response using these viral antigens which might be expressed on the surface. We'll see where it's common. Specific tumor antigens, as I told you, are identified in connection with different tumors, but you see, most of them are identified in melanoma tumor, but also on lung carcinoma, also in breast cancer. These are specific proteins, results usually of the mutation of the normal cells, so they are not found in the normal cells, and these are specific tumor antigens. I'm not going to give because they are different, and these are not all. A lot, any um, in moment the new antigens are trying to be identified because, as I told you, this tumor antigens is a very useful target for specific immunotherapy. And you will see a little bit more there. So, in what is the role, how this immunological we identify tumor antigens are used in oncology. They might be specific tumor antigens are used to identify and stage a different location or different types of tumor. Also, and this is done by molecular PCR reactions and so on. Immunophenotyping of leukocytes is also, we don't, you remember how many immune cells take part in surveillance. But and in some when we speak about reduced immunity in cancer, we should use we should test cellular and humoral immunity. So this might give you information of the um, state of the immunity during this. And here the important area of usage of these tumor antigens are so-called tumor markers, which are you meet in clinical practice. They are, as I told you, they are molecules, macromolecules, which might appear in normal, but in a very low concentration uh, cells, their amount in the serum or other liquids in the organisms are increased when there is a tumor process. That's why they are called tumor markers, but they are not only typical, they are not specific for the particular antigen. They are not specific tumor antigens. And these tumor markers are usually identified by the ELISA immunoenzyme methods or other immune enzyme methods. Nowadays, they are put on the automatic, and this technology is automated in immune analyzers, and they're very useful and they are very used in the clinical practice. But these tumor markers, as I told you, are not specific. They do not put your diagnosis, but they're important to follow up the development of the tumor process, the results of the therapy, because the amount in the blood, in the serum, it's easy to be tested with these auto analyzers, and they give you information if the therapy is radical or the therapy is not, and they should you will see a little bit more. What is important here to know that these are not specific, as I told you, for the certain tumor, but they do not give you a diagnosis, because you can see that in healthy population, about 20% of the person may have uh, low level increase, but low level of these tumor markers, and they do not have expressed disease, but they might have, because they have 20% might be positive for tumor markers. And the same is connected with the sensitivity, because 20% of the healthy person might express also. So here you 20% you miss these tumor markers, but you can make an overdiagnosis here in healthy because of the high sensitivity. That's why these tumor markers are not considered specific for certain diagnosis, but they work for the follow-up, as I told you. So if we have a case here, a case here with the, I think this was carcinoembryonal antigen, which is a tumor marker, and if this, you can follow up easy these concentrations in serum of the patients, and you can see that if there is an increase before operation, there is a possibility for three types of curve which might be. But in one case, in some patients, this tumor marker is effectively how to say, not expressed in the uh, peripheral blood in serum, and this might be under effective therapy or effective chemotherapy or even a surgical operation. Then, if it's not appeared, so it's considered that there is no residual disease or appearance de novo of the tumor. In some cases, it goes down under the normal level, and after sometimes it starts appearing increased again. So this is considered a situation when the new tumor or in the same in situ tumor is appear again. And in some patients, this tumor markers is not significantly reduced, which means the therapy is not effective and the tumor is still there. And there is several tumor markers which are most often used in the clinic. And gastroenterologists are using very often CA 19-9 tumor markers, and this is characteristic for the colorectal carcinoma, pancreatic, and stomach, and also liver carcinoma. The liver cells produce these tumor markers. As I told you, they might produce in healthy, but it is under a certain level which is uh, accepted as a reference level between health and pathological level over 37 to 70. The diagnostic value, as I told you, it's not specific, but mainly in pancreatic carcinoma is important, and it is important to know that the level of this tumor in the peripheral blood is in correlation with the uh, tumor growth, is with the volume of the tumor mass. And when it is a small one, there is no significant levels which could be connected. But in about 80% of the patients, was localized carcinoma when the levels are increased and the values are increased. As I told you, this marker goes well correlates with the volume of the tumor mass under 2 centimeters in that it might be negative, but with the growth of the tumor mass, it appears in a higher percentage of the patients is a positive. It's a good criteria for effectiveness of the surgery if such surgery operation was done, and it's a good marker for uh, therapeutic effects in the serum and do other liquids. So this marker is useful. It's very often used in gastroenterology, but as I told you, it is not specific for particular tumors because this tumor marker might be found in another diseases which are not carcinomas, such as toxic hepatitis. About 14% are positive. Chronic active hepatitis, up 33% are positive. There's no carcinoma here. Cirrhosis up to 10%. And as you see, uh, might another biliary carcinoma of biliary system has a very high incidence of these tumor markers. So that's why tumor markers in this one is not considered specific, and you cannot use it to prove the diagnosis. But they are very useful for the follow-up of the results of the therapy in such patients. Combination of this marker, CA99, with other tumor markers, so-called secondary markers, primary and secondary, increase significantly the percentage of detection or improvement of the carcinoma. So, pancreatic carcinoma in combination with 99, carcinoembryonic, and C100 and ferritin. This is the optimal combination for proving carcinoma, pancreatic carcinoma. For stomach, combination with carcinoembryonal antigenic. liver carcinomas, carcinoma brunal, and 90 then. What is important that combination with other markers usually increase the assurance that it's connected with the certain tumor. And this is valid for the other tumor markers also. If we look at the CA 125, this is characteristic tumor marker for ovarian carcinoma. For all healthy, it's very rare, low concentration. And here different type of tumors might be connected. 78%, about 80% of has diagnostic sensitivity, 90% specificity for epithelial carcinoma for this carcinoma. It's also depend on the level of incidence, depends on the size of the, here is on the stage of the tumor, but in the size as well, we tell you. And combination is very important also. And the main, as I told you, the main importance is for the follow-up. So for the tumor markers, what the algorithm we use usually, it's because it's for the most useful is for follow-up. So it's absolutely necessary to have tested to this tumor combination of tumor markers before the operation or before starting cytotoxic treatment. Then after a day, uh, 10 after the surgery or the beginning of the therapy should be tested to see the first results after beginning of the treatment. Then 1 month after the treatment with also with the cytotoxic or every month, the first 6 months should be followed up every 3 months after that if the situation goes uh, happily. So will make, but you need to make check on certain periods. And also this tumor marker combination with the other one increase the diagnostic use of this marker, of this tumor markers. The development of tumor markers of this tumor, it is the first slide. You see, you have a look on them and found this heterogeneity, a very big heterogeneity of the tumors, different type of tumor cells in one tumor location, as I want. Microenvironment and stem cells therapy is very important. And what we could conclude from all what we have until now, we proved that antigenic tumor antigens are not very immunogenic. And I show you why it's not considered not good recognition or presentation or suppression. The immune response against tumor antigens is slow. Take times until it appears. Cytotoxicity is, which I told you, this is the main process which kill the tumor cells, is a weak one in humans. The tumor cell itself make a very strong suppression onto immune responses, and I think I showed it to you. And specific immune responses in tumors is very difficult to enhance. That's why these are the reasons why the very, very important part of the therapy, instead of cytotoxicity or X-ray, we have increasing uh, importance of the immune therapy. That's why I will have some words about this. The aim of immune immunotherapy is to enhance the immune response in order to kill tumor cells. This is the main uh, aim of this therapy. Different approaches to this aim are made or are used in immunotherapy. The first one, the beginning, starts with non-specific activation of the immune system with common usually uh, vaccine. What is important that one of the beginning, in 50s and the previous age, about 1950s, the Bulgarian oncologist, economist, was one of the first which used non-specific activation of the immune system in order to induce effective immune response against tumor, and it used melanoma tumors and activate these things. Induction with viral antigens, it is good, interesting, but only for tumors when you know which antigens are is expressed there to induce antiviral immune response. And for humans, it's a Burkitt lymphoma and eventually carcinoma or cervix carcinoma with HPV, but it was expected more than 10 years now to produce some big pharmacy firms to produce therapeutic vaccine with HPV, but still not very popular. It's difficult to induce this response. Another approach is immunization with tumor-specific antigens. So these are viral antigens, these are tumor-specific antigens with peptides or with the gene, transfection with the genes for this identified tumor antigens. We'll see a little bit more then. The other way to increase the immunogenicity of the against the tumor is to uh, provide the missing chains. So in with usually transfection, genetic transfection with costimulatory molecules, as I told you, one of the mechanisms to energy to break the immunity was missing tumor cells do not express these costimulatory molecules. Transfection with the genes for MHC class 1 to improve CD4 recognition, presentation first and then recognition by CD4 and induction of cytotoxicity and immunization with the dendritic cells with tumor antigens. See a little bit more about this difference. So this is the area of, in this one, the area of so-called tumor vaccination or anti-tumor vaccine therapy. So enhancement of the non-specific enhancement, as I told you, this is usually in the beginning it was done with Calmet-Guerin, but nowadays it's different other ways of this non-specific activation of induced of immune response. And one of this is uh, vaccination or introduction of this bacterial BCG. This is Comet-Guerin, Corynebacterium parvum, which the idea is to activate ex vivo or in vitro these tumor cells and then introduced again in the organism. So it might be, so it, how it's done from operation to the surgery is taken out the tumor. Tumor cells were uh, prepared in a single cell, how to say, suspension, and they might be several ways activated. One is to incubate with this uh, bacteria and activate these tumor cells to produce or to express several mechanisms. The other one is to transfect it with the genes for cytokines or for which are needed to improve or to increase the immune response. And the idea is this manipulated tumor cells ex vivo or in vitro can be introduced again in the organism where we expect to have this improved immune specific, no, improved immune response. One other way to use this Calmette uh, preparation is to use it in a urine bladder where the carcinoma from for carcinoma bladder carcinoma is very good how to say treated with injection instillation of this tuberculin antigens into the bladder. So as a final result is activation of tumor in infiltrating lymphocytes. We uh, we don't have time. We did not speak about this, but tumor mass is infiltrated with the tumor cells, and these lymphocytes is not active. The action here is to make these tumor lymphocytes more immunogenic and make this effective cytotoxicity in these lymphocytes. Another way, another approach is to improve the cytokine secretion for different or use it as a injection or introduction of different cytokines such as interferon alpha was used in certain types of carcinomas, interleukin 2 or TNF alpha. All these cytokines are introduced into the patients with the carcinoma and expect to improve the immune response. But and here are several examples when interleukin 2 is introduced, it decreases the NK activity and so-called leukocytes activated killer cells. TNF alpha or interferon gamma also is introduced. But the bad news here is that these interleukins are very gives a very strong side effects with the temperature and with very heavy fever. Then these patients not very easy uh, take this therapy. And the other version of this cytokine. That's why nowadays there is a introduction of cytokines or stay in some location of the carcinoma, but not very often used. There is a possibility these cytokines to be introduced not as a proteins which induce this fever, but as a cytokine fever, but introduced as a genes for these cytokines, and the organism to itself produce these cytokines. Another induction of enhancement of the anti-tumor specific response is when tumor cells, like the first one, tumor cells are isolated ex vivo from a tumor mass which is removed, and by different approaches, these tumor cells themselves are activated by radiation, by destruction to aim to express more antigens by transfection from different molecules or cytokine or lysing these cells, and the aim is these cells then to be introduced again in the organism to enhance the immune response, cellular immune response. This approach is called adoptive transfer of these manipulated cells in human organism. Of course, these are very attractive approaches. They are mostly on experimental levels up to now, but they are expected to be, or a lot of effort is done because they are very clever as a approach to increase the immune cells. Um, Monoclonal antibodies were one of the very expected, uh, how to say, approaches to produce the anti-tumor cytotoxicity. That's why they were called in the beginning magic bullets. What is it? Monoclonal antibody is produced usually hybridomous in mouse. They might be humanized, but it's very expensive and it takes a long time. These monoclonals are directed against a certain antigens into the tumor cells. If they are loaded with the toxins or with radioactive uh, elements, and they are introduced in the serum, they will go and find the tumor and it is expected to make such cytotoxic effect of these additional structures, radio material or toxins. But here is here is the bad news. This, as I told you, tumor antigens are very quite unstable and they are very changeable. And this antigen uh, shedding or editing modulation is a characteristic. So until these antibodies are produced, monoclonal antibody, it takes months to produce, and when they introduce to the patients, it might be too late because this antigen might be the same antigen might be on not on the surface to be a target for this conjugate. That's why this method is very attractive, but it's not very effective and very expensive and it's very expensive to be used. So I think something happens with the slide. I don't know why it's not going. But I think these are the most I showed you the approaches in the immunotherapy. Of course, nowadays there is a new one. As I told you, this is a very uh, flourish area, very fast developing area for these therapies, and they are very looked for certain patients, but most of these approaches are still in how to say experimental research approach. Of course, there is a so-called target therapy, and it is in practice. They're quite expensive, as you know, but target therapy is a biological or therapeutical drugs which are how to use, which are directed against certain molecules or proteins on the tumor cells to induce activation or suppression of their activity. So these are the new approaches in tumor treatment. So I think I will finish now. Please, do you have questions? Are you there still?
Yes, doctor.
Do you have questions about the lecture? It's not very easy as a how to say as a topic, and because we do not have the initial lecture also for clinical immunology, but I'm still here. Please ask if you don't understand something or you're interested in something else. Will not have very much lectures on the tumor immunology in other disciplines. Do you have questions?
I think everything is clear.
Okay, it is good. But may I ask you? So we lost first three lectures from our course. You have one more week. You finish your practicals this week. Is it right? Am I right?
Yes, we will finish this week.
Yes, this week. Next week, the Monday is free. You don't have other lecture at the same time. Do you want to have another lecture then? What I can propose to you, we would have primary and secondary immune deficiency diseases. But I don't know because it's a week before Easter. I don't know. Maybe are you going to be here? Do you want to have this lecture? I'm ready to present it. Will you answer me
Yes,
now?
I think we can have another lecture.
Sorry, what?
I think we can have another lecture.
So I could have another lecture next week. Yes, with you.
Yes.
Okay, same time next week we will speak about inherited immune deficiency diseases and acquired one. Okay?
Okay, Professor.
Okay. Have a nice day. Thank you for your attention. If you don't have any more questions, we finish today. Okay?
Okay, thank you very much.
Thank you for your attention.
You are currently the only person in this conference.